This video is going to be an introduction to chromosomes. As mentioned in our introduction to division video, one of the important things that we need to have copied before dividing is DNA. And of course, we've talked about DNA being only in the nucleus, and so you need to know that when DNA is in the nucleus, it is in the form of chromatin. It isn't necessarily packaged when we think of packaged like chromosomes. It isn't packaged at all. It's all tangled up. In fact, if I were to draw a nucleus here with some chromatin in it, it would look like this. I mean, it looks like a plate of spaghetti. It's all tangled and things like that. Okay. During division, we pack up the DNA. We do this because it'll be easier to move pieces of DNA around and for the cells to check that every new cell has exactly the right amount of DNA. In prokaryotes, prokaryotes do not have a nucleus. Their DNA usually is in a particular region of the cell called the nucleoid. And most prokaryotes only have one circular DNA chromosome, so it has basically one piece of DNA, and it is in the form of a circle. That basically has almost all of the cell's DNA. And as mentioned before, a chromosome is basically a packaged version of DNA. Eukaryotes, on the other hand, have lots more DNA than prokaryotes do. Obviously, we are way more complex than they are and need more DNA and more genes. Not only do we have more DNA, it exists in the form of multiple chromosomes. So whereas prokaryotes have one chromosome, eukaryotes will have multiple chromosomes. Eukaryote chromosomes exist in sort of line shapes like this, or sort of singular like that. They're sort of linear. And the chromatin will condense to form a chromosome in this way. So you can think of our DNA double helix, which we learned about in a previous unit. It gets coiled around various proteins and then coiled again. Look at, we call these supercoils, and those get coiled even further to basically make a chromosome. I like to think of a chromosome like a sock, and the DNA is like all the little threads that make up your sock. Okay? Way easier to sort your socks if it's in sock form and not in threads. That would be miserable. So again, chromosomes make it easier to divide the DNA properly during cell division. Just some chromosome terminology here. Usually an unduplicated, so uncopied, chromosome looks like this, and we can draw a replicated, or we can use the word replicated or duplicated chromosome as such to the right. You should note that cutting it down the middle like that gives you both of these. So again, if we go through DNA replication, we now have a double set of our DNA, and that's what it looks like. Some terminology. This central portion of the chromosome is called the centromere. Chromatids are what we use to define the two portions of a copied chromosome. Okay, so we have one sister chromatid and the other sister chromatid, and because they are of the exact same chromosome after DNA replication, they are identical. And they are linked together at the centromere. One big thing you have to notice is that both of these images are considered a single chromosome. We can refer to this whole thing as one chromosome made of two sister chromatids, or one chromosome here of a single chromatid. We will see something like this after a duplicated chromosome splits. So again, just some definitions. A centromere is the central place on the chromosome where sister chromatids attach to each other, and a chromatid, as in saying sister chromatids, is each strand of a duplicated chromosome.